So, bacterial metabolism and genetics, that's the topic for today. And those of you who have never seen a virus, this is how a virus looks like. And this particular virus, this is important to infect bacteria, is called bacteriophage. So you can see that not only human beings get infected, but bacteria get infected by virus. Today we'll talk about the growth of bacteria. I'm going to talk about the requirements, physical and chemical, for growth of bacteria, because obviously we want to stop that. I'll also talk about some of the genetics of bacteria as well, and how does bacteria transfer genes. Now, a little bit of recap from your biology. You must have heard many a times how a cell divides, and pretty much the same story. Cell elongates, DNA is replicated, then you see a groove there, and eventually one cell divides into two daughter cells, and so on and so forth. So that pretty much is a situation for many a bacteria. And in this case, you can see, depends upon the rapidity with which the bacteria divide. But also keep in mind that this division of bacteria, when one cell splits into two, is a very coordinated process. It's not going to need energy, it's going to need nutrients, and by the end of the day, if you run out of nutrients, for example, the bacteria will stop dividing. So they are like physical and chemical factors. You have to keep that in mind. And the other important thing is that there is a nuclear material, a genetic material that's been divided. So the daughters share the mother's genes, so to speak. Now, also keep in mind that we will talk about some of the regulatory events in the synthesis of proteins and RNA in this case. But one thing you have to keep in mind, just as a concept, is that once the signal is delivered to DNA, and the cycle starts, and then it has to complete. Right? So cycle has to complete, and also keep in mind that even if all the nutrients have been removed from the media. So that's a key point that I will spend some time on today as well. Now, if you looked at the previous slide, you have a pretty much idea about the structure of bacteria. We talked about bacterial membrane. We talked about peptidoglycan synthesis, and most of you will spend quite a lot of time today in the lab. And that's the outermost layer for both gram-positive and gram-negative. Gram-positive have a thicker layer as compared to gram-negative. And also keep in mind that ideally, if there is one staph aureus, and if it divides into two, they are the same staph aureus as the parent, as the mother. But sometimes it may happen that there is a resistant strain developed. So where is this resistance coming from? Why all of a sudden we have a problem with antibiotics? So that's something that you have to keep in mind. Now, as far as bacterial cell is concerned, also keep in mind that the genetic material that bacteria has, though it looks a little bit scattered, there is no nucleus per se, but even then, there is a very precise division of the genetic material, and cell basically divides into two. There is a multiplication time we call dividing time. So many bacteria may have different dividing times. So if you were to plot a graph, for example, if I was to plot a graph in terms of the log of number of bacteria versus time in hours, that's the dividing time. So you can see it goes through 1, 2, 3, 4 definitive phases. First phase we call lag phase, log phase, stationary phase, and then death, basically when there is a decline in the number of bacteria. Why do we have lag phase? Because at that time there is an intense activity where bacteria is preparing for all the nutrients required for it to divide. And then again, once you have that, there is a logarithmic or exponential increase. You can see log phase. And then again, there will be a time, a natural curve, where you will have a stationary phase, and then there is a decline phase. So it's pretty much key concept for you to have that all bacteria have to pass through these phases. Not to tell you that bacteria, even when they die, they are any better than or any worse than they are alive, because if they have an outer covering endotoxin, they will still cause the damage. So the point to have to keep in mind in this case is that when we go to that exponential or log phase, there is an increase in cell number. So if you were to come up with a drug, or you were to come up with antibiotics, you have to make sure that you hit this phase, because we do not want bacteria to divide, correct? Okay. Now, also keep in mind that you have to ask yourself that why do you refrigerate the food? Why do you freeze the food? And the answer will be because you don't want bacteria to multiply in your food. So you want to keep them for a short time in the fridge. You want to keep them for a longer time in freezer, or you want to keep them into deep freezer for even longer time. So what you are doing is that you are not providing an optimum temperature for bacteria to divide. So of course, these are important. So many a time you may have heard grandmother's way of cooking, where you increase salt, increase oil, increase sweet to preserve food. That again basically is coming from that idea that the bacteria do take influence for all these physical and chemical gradients. So we do see that temperature is important, pH is important, osmotic pressure is important. And remember, I told you the spores cannot be killed unless you autoclave your clothes or your gowns that you wear. Now, when we talk of temperature, bacteria, for example, if you were to leave food on desk, on the table, or leave food in the refrigerator, or leave food in the freezers, you will see that there's going to be a proportional change in time of decaying food. The chances are you're going to leave it overnight, it may get decayed on the kitchen table earlier than the fridge. So there is a definitive influence of temperature. So if you were to look at generally, you will see over here most of our body temperature, most of the temperature that we are working with is the temperature where we have rapid growth of bacteria. It's called danger zone. And then again, if you go up, you can boil, you get rid of bacteria, or you can go down and you can basically stop that progress of bacteria. pH is important. I talked about the pH in the stomach. Acidic pH that will stop bacterial growth. We talked about different flora. I talked about vaginal flora where you have acidic pH. Then again, will stop multiplicative progress of many bacteria that may be harmful to that environment there. If you look at osmotic pressure as well, many times you must have heard doctors saying that, put a salt in the water and do gargling, right? So that's good for your sore throat. So you can see that the whole idea for these things is that you can destroy bacteria if you change the hypertonicity of the environment, because they are bound by a cell wall. They have a cell membrane. If there is a change in the ion concentration, of course, it's going to either shrink or swell the wall of that. Bacteria, of course, has chemical requirements, and there are many things that we see in terms of bacterial growth. We have nitrogen, sulfur, and phosphorus, and you can see many times when we get specific bacterial infections, our bodies get depleted of these particular chemicals. So if you look, for example, for some of the levels of, let's say over here, a particular amino acid. So that amino acid is deficient, like thiamine or biotin. because the infection in your body is consuming for the growth of bacteria. So that's the concept people come up with, that when you have infection and you're taking antibiotics, you may want to have complements or supplement in terms of multivitamins and so on and so forth, but that's true as well. If you look at trace elements, and then there are many enzyme cofactors, because the whole idea is that bacteria is dividing at your expense. So it's basically taking your nutrition requirements and dividing it in number. So we will run short of those particular trace elements that we have. The most important that I have discussed earlier as well is the requirement for oxygen. So you can see there are many ways of dividing bacteria, but this is an easier way. And then again, also keep in mind that some of them basically will only live when they have oxygen. So they basically are called obligate aerobes, as compared to some of them basically they're going to run away from oxygen. They don't want to have oxygen, so they are called obligate anaerobes. And then in between they are facultative. They are those bacteria, they can make do, whichever situation you give it to them, either oxygen or no oxygen, they can make do with that. But at the next time, there are some other ones, they may have very specific requirement for oxygen. Right? So as I said earlier, most likely respiratory tract infections are caused by those bacteria, they want to grow in oxygen. As compared to wound infections, for example, where there is no oxygen, there is a dead tissue, some of the bacteria want to thrive on that. So that basically is coming from some of the enzymes that these bacteria have, and these are important. So if you pay attention over here, why is it that this bacteria wants to live in oxygen? Because it has those enzymes which are catalase and superoxide dismutase, and they allow toxic form of oxygen to be neutralized. So they have this capability to neutralize toxic form of oxygen and make use for them, as compared to the other one, which also have catalase and superoxide dismutase, and they can basically neutralize the toxicity of oxygen, right? And then some lack this enzyme. So these bacteria do not have those enzymes. It's a very common question asked. And then the whole idea is that bacteria themselves carry these enzymes, catalase, superoxide dismutase. And the whole idea is that these enzymes are going to chew up the toxic radical and then make them survive. Okay, and then you can see some of the other bacteria may have specific need. I'll give you an example, like Clostridium, for example. So Clostridium tetani, this is the one that you get by roadside accident. And this is a bacteria that doesn't like oxygen. It wants to thrive on dead tissue. So when there was an outbreak, or there's a big epidemic of that particular bacteria or any other bacteria that wants to grow on dead tissue, so they want to treat, they used to treat the patients by putting them in oxygen chambers. So they will put them in oxygen chamber and treat the whole body, hoping that oxygen is going to kill those bacteria, because those bacteria don't like oxygen. So that's the whole point over here. So use of oxygen, and then also important for many different things. For example, antioxidants. We talked about antioxidant. And also remember, I told you when the neutrophils, they phagocytose the bacteria, the killing enzyme they have is basically reactive oxygen intermediates or reactive nitrogen intermediates. So they have those, so to speak, bleaches. It's like hydrogen peroxide. So hydrogen peroxide that you normally use, that is basically used for wound cleansing, because it is going to cause damage to the bacteria, right? And it is a bleach as well. Okay? Now, When we talk of bacterial genetics, some of the key concepts, again, go back to that basic table that you saw, differences between eukaryote and prokaryote. How is it that our nuclear material is different than the bacterial nuclear material? So bacterial genome has a single haploid chromosome. That's the most important thing. It has a single haploid chromosome. And what other things that it has? Some other genetic elements which are extra chromosomal pieces of genetic material like plasmids. And I'll talk about bacteriophages. And what are bacteriophages? Virus that infects bacteria. That's what the key first slide was. Now also, remember, the question you have to ask yourself to understand today's lecture is that why do we get drug-resistant bacteria? If you say they go and divide by mitosis, by binary fission, so mother, daughter, so where is this extra piece of information is coming from? So that is basically a Nobel Prize winning research. So let's talk about some of the important genetic elements of bacteria. So what are bacterial genes? Right? Because bacteria has protein on the outer side, it makes glycoproteins, and it has many different lipid parts. So it is obviously manufacturing through the genes. Now, we human beings, we have two distinct copies of each chromosome. Therefore, we are called diploid. So we are diploid as compared to only one copy of chromosome which the bacteria have, and these are haploids. Now ask yourself this question: if that is true, so bacteria only have one chromosome, if by chance there is an alteration of gene which is called mutation, it will have an obvious effect on that particular bacteria as compared to human beings. So if you look at the probability or the chances, your statistics, right? If there's only one and something's going wrong with that, the chances are it's going to show up. The other important thing, those of you who are interested in DNA structure or RNA structure, that if you look at this one, for example, a typical cell, and of course this is not a bacterial cell because it has a nucleus, and you can see they have a chromosome, and if you open up the chromosome, like open the thread, and you will see a DNA, but DNA is bound into that specific configuration by something we call histone. So histones are there. But of course, bacteria would not have histones. Okay? Now, remember, as I said earlier as well, whenever bacterial DNA is dividing, we hope that nothing should happen, will happen, but cell has a machinery to check that there should not be any mutation. If there are a mutation, they need to be repaired. So that's the whole idea. So there's a system over here that we want to make sure that there is an accurate replication of DNA. So that's the key over here. And you'll ask yourself, why is it important for bacteria to make sure that bacteria has the accurate replication of RNA or DNA? What do you think? Why is it in the interest of bacteria that it has to have a accurate replication of DNA? Why? Survival, of course, because if it doesn't have that, it will change. That's the whole idea. For bacterial survival. But again, having said that, mistakes and accidental damage to DNA does occur. So that's the key over here. That though they have a repair system, if there occurs some mutations or alteration to DNA, things can go wrong. Now, the other question you will ask yourself that, of course, the prevalent hypothesis is that, for example, just pick up E. coli. So E. coli has been there ever since we know E. coli was there. So it hasn't kind of mutated to such an extent that, like viruses, you have every year a new. If had it been the case, then we would have been in trouble. But the question you may ask yourself is that ever since we started using antibiotics, so antibiotics were kind of discovered maybe like 40s and 50s, 1940 and 50, or maybe started using in 60s. So it's like maybe half a decade. So ever since we have started exposing these prokaryotes to antibiotics, they are also coming up with techniques to resist it, to avoid it for their survival. If you compare that with viruses, for example, the general body of agreement is that there should not be any smallpox virus in the world, hopefully, except in the labs. There should not be any polio virus in the world, or plague virus, so a plague bacteria. So the chances are that we pretty much have eliminated that. But that is not happening even with our use of antibiotics, that they are still there. They keep on coming over and over again. So the idea in this case is that some of these mutations that happen in bacterial genetic transfer is beneficial. It may improve the chances of survival. So that's where it is coming from. Now we need to learn and find out that how does gene exchange takes place in bacteria. Especially we are interested in pathogenic bacterial species because they are more dangerous. And the idea is that when there is a exchange of DNA happening between two species of the same kind, and one becomes pathogenic, the other is non-pathogenic. So what is this transfer of information that makes one pathogenic? So what is happening in terms of development of a new strain? So what we have found is that basically it has worked to the advantage of bacteria, and the information that bacteria passes from mother to daughter, how to avoid antibiotics, how to resist. So there are some DNA codes. So there are some DNA information and code that is passed. So that was a big question for the scientists. So if the bacteria divides into 2 and 2 and 4, how and why and where is this information coming out? So in the last few decades, they found out that those little tiny extra chromosomal pieces of DNA that we used to call plasmids, and they are like double-stranded DNA molecules, and they are circular, and they have about like 1,500 to 500,000 base pairs. They are the one that carry that particular genetic information, which is not essential for the bacteria for survival, but it's an advantage for the bacteria to pass some information in terms of antibiotic resistance. So plasmids basically was the #1 causative organisms for bacteria. Number 2 is, when we did research on plasmid, we found out that not only the antibiotic resistance comes from plasmids, but some other pathogenic factors or virulence factors or violence determinants that will make one bacteria pathogenic as compared to non-pathogenic is also being transferred through those extra chromosomal pieces of information called plasmids. Now, let's talk a little bit of plasmids, because plasmids again are important. And right now we have different types of plasmid. I'll just talk about two plasmids that are important. One is called conjugative plasmid. So this is a plasmid that basically when bacteria passes this genetic information. Remember, now I'm talking about a little bit different. We're not talking about the binary fission. We're not talking about the mother-daughter split in terms of one bacteria dividing into two. We are talking about some extra, we call it other factors for genetic transfer, like promiscuity, so to speak, for bacteria, and especially for pathogenic bacteria. So they want to invest, they want to use their bacterial DNA for a particular purpose. So one purpose is that these bacteria will conjugate with another bacteria. So this conjugation of bacteria will transfer via sex pili from so-called male bacteria to a female bacteria, where this information is passed on. So one of the ways that bacterial resistance or antibiotic resistance is going to be transferred is conjugation. Okay? And you can see from here, when this kind of information passes from one bacteria to another, or a code that passes from one bacteria in O to another bacteria, so that particular information is going through a very defined genetic material. And if you look at these plasmids over here, this is a electron microscopic, so double-stranded DNA, you can see from here. And then they are circular. And then there are different protein sequences over here. So they have different kind of details that we have, how to transfer and what to transfer and so on and so forth. The example for this kind of conjugation is that plasmid is that we can basically use it for our own industrial use as well. So if you want a particular sequence to be developed, so you would insert into this plasmid and infect the bacteria with this plasmid. And then bacteria will start making this up. So this is many times we produce many proteins, industrial use, by our capability to go and open up these plasmid and add the genes of our interests. Okay? The other important, so conjugation, when bacteria mates, so to speak, with another bacteria and passes this information for genetic transfer of antibiotic resistance. The other important thing is bacterial viruses. So what happens is that there are two bacteria. One bacteria gets infected with a virus, and this virus will put the genetic material into that particular bacteria, and this bacteria then will divide or used as a parasite for replication of viral DNA or RNA. So in the process of doing it, it may have those extra chromosomal plasmids. So they will also get incorporated into viral genetic material. And when that cell disrupts, it picks up that virus, and that virus has these plasmids. And then plasmids will go and infect another bacteria. So they basically will be used as a conduit or a vector to take one particular genetic material from one bacteria to another bacteria, right? And then again, this basically happens in two ways. One way is that the virus will go and infect, and then the virus will divide in bacteria like any other cell, and then those viruses are released open without the lysis of bacteria. So this basically we call a state where bacterial cell itself is not broken up. It's not lytic, so they can survive. The other important thing is that they can go and divide into this bacteria and then break those bacteria apart and then be released. So these are the two major ways that we normally see. Third type of things that we normally see where bacterial DNA can transfer from one bacteria to another bacteria are transposons. So these transposons are called jumping genes. So these kind of those DNA material that is out there, and then basically it can go from one bacteria to another bacteria, from one cell to another cell. So this is another important thing that you can basically encode. So you can basically take one gene, insert the gene of your interest, and put it back into the genetic material. Okay? So these are some of the ways that we normally think they are there. Now what we want you to do is that again try to understand this concept, because this will be the only time that we discuss it. And the next time we will ask you to give us reasoning, where is this antibiotic resistance coming from, and what makes a bacteria resistant to antibiotics. So you have to keep in mind that basically it is coming from the bacterial genetic exchange. So that's the whole idea. Bacteria are passing those special genes to their progeny, and I think some of the books mention 4, but your book, Murray's, has mentioned 3, and I'll talk about why is it 4 missing there. But if you were to remember these 3, number 1 is in conjugation. Bacteria mate with each other via sex pili. The second one is a transformation. I'm going to talk about that by some experimental things. And the third one is a transduction. So these are conjugation, transformation, and transduction, and we'll have a full workshop on that as well. So let's talk about conjugation in a little bit detail. And I think you pretty much got the essence of it, that what we are trying to say is that the bacteria, which basically are in the same species or related species, they physically come in contact with each other and they conjugate. And there is a bridge, which is in bacterial case we call pili. And this particular pili, because it transfers genetic material, is called sex pili. So in this case, there is a donor and a recipient, right? Or a male or a female. So pretty much the same conjugation that happens in other animals. Now, if you look at this one, the particular plasmid that is transferred by conjugation has a factor. This is called factor, and many times we call it fertility factor, F factor. So a bacteria which will have that factor we call F positive, and we also call it male. And the bacteria which does not have that factor, fertility factor, we call female. So that's like F positive, F negative. So what happens is that the donor will transfer the whole pieces of chromosome into recipient bacteria. So that's the whole idea. Okay? And this one specifically, when it happens in terms of a particular plasmid, we call that as a conjugative R factor. So that's important. So let me actually show you the figures, and you, those of you who have read it before me, may have already seen that. Okay, there you go. So if you pay attention over here, so far I mentioned why do you get bacterial antibiotic resistance? Because the information is passed from one bacteria to another bacteria to encode for those specific resistant genes. And that information is in plasmid. Now this is one bacteria, you can see from here. This bacteria has this is a typical bacterial chromosome. And then it has extra chromosomal material, which I told you is a plasmid. And this plasmid has a F factor. Okay, so we call it F positive bacteria, F positive cell, or a male cell. And then basically it attracts a female cell, which is F negative cell, then establish a conjugation by a bridge we call sex pilus, and this one is transferred, the whole plasmid is transferred there, and there is a replication of the plasmid. And then you will see by the end of the day, we'll have 2 F positive cells. Does that make sense? So the gene for antibiotic resistance is transferred from one bacteria to another bacteria by a process known as conjugation, where F positive is transferred and replicated into F negative cell, and then both of them will become F positive cell. Okay, this is one way. Let's talk of another way. So there's another way of conjugation. Now in this case, what you normally see is that, again, there's an F positive cell, and it has a bacterial chromosome, and it has a plasmid over here. But what it is doing is that instead of passing it through conjugation directly, this is going to get integrated into bacterial chromosome. So when such a thing happens, where there is an insertion of F factor into chromosome, we come up with the integrated F factor, and this particular cell is called high frequency recombination. We call HFR, high frequency recombination cell. And again, the same information is passed via conjugation for bacterial resistance. Okay, so these are like 2 ways. Now, the third important thing is that now you have a cell which is high frequency recombination site, where the F plasmid is a part of bacterial chromosome. It can also then again conjugate via a bridge we call sex pili, and then this particular HFR is basically replicated into the recipient cell or bacteria. Okay? And in this case, you can see again that in this kind of recipient recombination, HFR chromosome fragments and the F chromosome into it, and then you basically have either HFR cell that you have to begin with, and then you have a recombinant F cell. So these are some of the ways that bacteria have developed, whereby they can pass this information to other bacteria to become, that's why they say that the bacteria in the hospitals are resistant strains. This strains because of this information of transfer of genetic material is happening over there. Okay? Now, the second process is called transformation. So what happens is, in this case, bacteria will pick up naked DNA and incorporate them into their genome. And if it happens to be homologous with the recipient chromosome, we call it homologous recombination. And that typically happens in these species. And again, this is a very common question asked. Name the bacteria that use a process of transformation to develop antibiotic resistance by a process of transformation, where they pick up the naked DNA from outside and incorporate into their DNA. And these are H. influenza, S. pneumoniae, and Bacillus species, and even Neisseria. So these bacteria have this capability of, that's what we call promiscuity. So they're going to take DNA, a bacteria lysis, bacteria breaks down, releases DNA, and these will pick up this DNA and incorporate into them. So that's another way of them acquiring it. And there was a very good experiment, if you pay attention, I think you will understand it for good. So what's happening over here is that when they gave a live encapsulated, meaning with capsule, bacteria into a mouse, so this is a capsule over here, and capsule is dangerous, and the mouse dies. And if you want to look at the colonies from the dead mouse, you can pretty much isolate the encapsulated bacteria because they are the causative agents. Now what you did was you took living non-encapsulated, so you put non-encapsulated bacteria into the mouse and the mouse remains healthy. It gives you that information that the lethal factor or the virulence factor is the capsule. That's what it shows you, right? So if you remove the capsule, the mouse remains healthy. What they did in the next experiment was they basically heat killed this bacteria over here. So heat killed bacteria and then inserted into the mouse. Again, mouse is okay, nothing has happened. Again shows you the problem basically is coming from the capsule of the bacteria. So what clever experiment they did was then they put both of them together, capsulated and heat killed, non-capsulated living and heat killed capsulated. And when they put them into the mouse died. Showing to you by some way the non-capsulated bacteria basically acquired, they transformed some information which gave them this capability to cause death. Okay, so this means is that they are the same species of bacteria. One is capsule, another doesn't have a capsule. If you put them together, they come close to each other. They can conjugate, they can transform, they can pick up bacteria and incorporate into them, and they become dangerous. So you can see living non-capsulated bacteria and heat kill injected, and then you basically are recovering encapsulated bacteria. The question you are asking yourself, you basically thought that you heat killed the bacteria over here. None of them have capsule. But the whole idea was that this bacteria passed some genetic information to the other bacteria to develop a capsule, and which was dangerous. Okay? So conjugation, transformation. And the third important thing where that kind of gene transfer takes place is called transduction. Again, pretty simple. A bacteria gets infected by a virus, and the virus had already picked up a resistant gene by infecting another bacteria which was already there. So it brought up one resistant gene from one bacteria and inserted it into the other bacteria. So that's what it did. So that's again important. And if you look at the whole mechanism of transduction mechanism, I think let me show you the picture, and this basically is a legend for the picture, so it'll make sense to you. So let's see over here, this is a bacteria and it gets infected by a virus. So virus comes over here, injects the DNA into this bacteria, and this DNA basically incorporates into the bacterial DNA, and then again you can see from here that once the virus uses the machinery of bacteria to replicate, it will basically have both. It will have some component of the viral DNA and will also have a component of the other DNA. So you have a donor bacterial DNA and recipient bacterial DNA both come together. And when the new viruses are made, so these new viruses, now which we call bacteriophages, will have DNA of the virus and the DNA of the bacteria. Correct? And if this happens to be the case in terms of this particular bacteria having those resistant gene, you can pass it on. So that was another concept that you have to have in terms of transduction mechanism for many bacteria for genetic transfers. Now, as I said earlier, this kind of recombination can happen we call it homologous, where the sequences of DNA are pretty much the same, or it can be non-homologous when the sequences basically are dissimilar, and you may basically insert a particular type of a gene into that. And that happens for some of the bacteria. The typical example you can see from here, that you have a bacterial cell. It has a chromosome A, B, C, D, and then DNA is out from the periphery, and this bacteria has a capability to take it in. Now, if these sequences are similar, we call homologous. If they are not similar, we call non-homologous. But again, you can see that this will go and combine and integrate into the DNA. Some of it will get integrated. So that kind of a integration where bacteria can pick up, we call it naked DNA, lying around from dead bacteria, and then insert it into their own DNA. And that kind of things happen at the level of genes. You can see chromosome A, exchange of genes between two DNA molecules. The crossover happens, and then you pretty much can transfer that DNA into the other one. The classical example that you will see in your pharmacy study, we call it vancomycin-resistant enterococcus, VRE. And in this case, Staphylococcus aureus has become methicillin and vancomycin-resistant Staph aureus. Staph aureus, as we discussed, is one of the notorious bacteria, and you can see that if we have two major drug of choice in terms of antibiotics, and this will become resistant to that. And again, you will see the way it becomes resistant, and it becomes like MRSA, MRSA, methicillin-resistant Staph aureus. The process of that basically is transduction and transformation. And again, you will see pretty much the same mechanism. It has to be either one or both mechanism in terms of how the genetic material is being transferred. Okay? Now, another important thing that you will see in this case is that if you were to isolate, especially vancomycin-resistant enterococci, in this case, this is another bacteria, you will see that most of this information is coming from plasmids. So most of this information that you normally see is coming from plasmid, and then by the end of the day, that pathogenic strain that carries that information has to be transferred. I gave you an example. You must have heard that the Staph aureus in the hospital, because there's a lot of antibiotics being used, and if it's not a good hospital, or even if it's a good hospital, the chances are that you may pick up MRSA. So if you pick up a MRSA, then antibiotics are not working against that. So that's a very serious problem. But we want to not only develop drugs, but we want to find out that how come a particular drug has developed that resistance. So that resistance, again, is coming from all those three important techniques that I mentioned earlier. And I encourage you to read it up and make sure that you understand, at least in terms of some of the bacteria, especially MRSA. And I repeat that the three major mechanisms are conjugation, right? And transformation, that you saw the experiment, the mice experiment. And what was the third one? Transduction. So these are the major things. And then again, once you come for the workshop, we want you to remember the name of bacteria as well. Which bacteria basically becomes resistant because of what mechanism. So you really have to associate. As I gave an example, for example, Neisseria. So Neisseria is important because it is bacteria's capability of picking up naked DNA from the environment. So these are some of the important things that are there, and please try to read it up. And again, I will also expect you to prepare for the workshop, but I believe workshop is maybe later in April, because this is something that will come over and over again. Okay? Any